Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be talking about DNA biotechnology. Now, of course, technology is the application of scientific knowledge to a practical purpose. So biotechnology is going to be the same thing, but with biological knowledge, with the things that we've discovered about biology. DNA biotechnology has had applications ranging from from gene therapy to forensic science, even to agriculture. So obviously, this is going to play a big role in our industry and in our technology. So that's what we're going to be talking about today. One of the first major advances that we had with DNA biotechnology was the discovery of bacterial plasmids. Now, plasmids are basically just little small loops of DNA that occur in bacteria. Bacteria have their main chromosome, which is just one long loop, and but they also have small plasmids. And the interesting thing about plasmids is that they can actually transfer them between different bacteria. So that's actually going to play a role in how we use them. Also in bacteria, we found what are known as restriction enzymes. These were actually used by bacteria to fight viruses and other kinds of infections. But essentially what they do is they cut apart DNA at specific places. So you can kind of see in this diagram, there's a very specific sequence in the DNA, and it will just cleave it right at that point, at specific points in that sequence. And so, using those restriction enzymes, we can cut apart plasmids and then insert sections of the genes that we want to insert, and those genes can be added to those plasmids. Then we can put the whole plasmid back into the bacteria, and then the bacteria will just reproduce as normal. After we do all this, we can get multiple copies of the gene, and now we have more to work with. And we have bacteria that may be able to express the gene, and we can have different ways that we can study the gene and its protein products. And so that really helped us advance it to where we can do more studying and more learning about bacteria, about the genes and the DNA. One of the next major advances was known as polymerase chain reaction, or PCR. I'll be referring to it as PCR. And the great thing about this is that it does the same thing as allowing bacteria to reproduce, but it amplifies the DNA in vitro, or in a test tube. That's what in vitro means. Essentially, they took a DNA polymerase from a bacteria that was found in a hot spring. So this bacteria was thriving at very hot temperatures, and because of this, the DNA polymerase won't denature when you heat it up, and that's part of the process in PCR. Essentially, each cycle of PCR is going to double the number of copies. So after you do maybe 30 or 40 cycles, you can have billions of copies of that DNA, and then you can insert it into plasmids to produce the protein, or you can test it to see what the sequence is, or whatever you're doing. But this PCR allows you to get all those copies of it, which is why it's so great. There are 3 phases to each cycle. First, denaturation, where you heat the DNA in order to separate the strands. What this does is when there's enough heat, the hydrogen bonds can no longer form, and so they'll just separate. This is known as melting the DNA, or denaturing the DNA, either one. Next, you have the annealing phase, which is essentially just cooling down the DNA, but you insert a RNA primer, similar to the primer that's used in DNA replication. And this primer has a very specific sequence. It's 15 or so nucleotides long, and so it'll bind to a specific part of the DNA and then allow it to be replicated from that point forward. And that third phase, which is known as extension, is where it's being replicated. And so this is where that DNA polymerase comes into play. And essentially, you just give enough time for DNA polymerase to work. There's going to be ATP in the test tube with it, and so that extension phase will allow it to extend forward. If you see this diagram, you start with the normal DNA molecule, which was shown in purple. this will actually have 2 primers, and so after the first cycle, you'll have a DNA with most of it replicated, and then as it goes on to the next phase, you get smaller and smaller. At the 4th phase of replication, you'll have two DNA molecules that are just the part of the molecule that you want, just the gene maybe, or the set of genes, just the region between the 2 primers, and then each cycle past that will double that number, or actually will more than double that. But after many cycles, you'll have almost all of just that little section that you want between the 2 primers, and so PCR is great for studying just that little snippet of the genome of the DNA, and this is how that works. Each cycle will double the number. Once you have that DNA, there's a process called gel electrophoresis that was developed to actually use to study the DNA. And what gel electrophoresis does is it sorts the segments of DNA by their size, by their length. Essentially, you have this electrophoresis tray, and inside of it is a plate of agarose gel, which is basically just this polysaccharide filter sort of thing. It's a gel. It's the same stuff that you have in a petri dish, if you've ever seen those. And it has these 3 little grooves where you can put the DNA in. And so the DNA is put into that tray, into those grooves, and then a current is actually run through the whole tray. And you can see you'll have the anode and the cathode, and because DNA has a charge, it has those phosphates on the side chain, so it has a negative charge, and when you run a current through it, it'll go towards the positive end, and so that force will be pulling the DNA through, but a force proportional to its size will be holding it back because of that agarose gel. And so the smaller pieces of DNA will be able to quickly travel through the gel towards the positive end, whereas the larger pieces of DNA will be held back and will travel much slower. If you have a proper DNA sample, you'll actually get these handy bands that show just the length, the relative length of those segments that are in that little part. And you'll actually have to use some sort of dye that will bind to the DNA in order to actually see the bands, but the DNA is there. If you have the other interesting thing is that if you have large amounts of DNA, such as if you're using an entire eukaryotic chromosome and doing that, it'll give large smears rather than discrete bands. And so this is kind of an idealized result. Also, one thing I failed to mention was that you would also use those restriction enzymes in order to cut whatever your DNA sample is into pieces in order to then use those pieces in the gel electrophoresis. So that's how you would get more than just one band. You would have a restriction enzyme cut your sample into pieces. Again, like I said, this is a very idealized result with only like 4 bands. But there's another process that we're going to get into in just a minute. That's how you can then distinguish if you have like a large smear of DNA. This next process is how you can then study that further. And this is known as southern blotting. Southern was the last name of the guy who developed this process, so not as in towards the south, but so southern blotting. And first you use a gel electrophoresis and you get whatever your result is. Often this will be with the whole chromosome, so it's hard to really tell where those discrete bands are, and so that's what this process is for. First, DNA is transferred to what's known as a nitrocellulose membrane, so just a certain membrane. This process also works with nylon, I believe, and so it's just transferred to that other membrane rather than the agarose gel. And then from there, you get probes, DNA probes similar to those primers that are complementary to the sequence you want. So these complementary probes, and you'll have them be radioactive, maybe with a radioactive isotope of nitrogen or something else. And these will be complementary to the sequence that you want to study. And so then these will be added to that nitrocellulose membrane, and will bind to the DNA in just the place where that sequence is found. Again, this will be something very similar to the primer, like I mentioned from PCR, where it's very specific to that one sequence. Maybe it's 15 or so base pairs long, so that it will only bind to that one place. But once those complementary probes have bound to the DNA, you can then use a film that can see X-rays or gamma rays or whatever to detect where the bands are. So that'll detect the radiation, and then when you look at the film, it'll show you exactly where just those 4 bands are, like with this example, where just those 3 or 4 bands are, and that will help you study just the small sequence that you want to study. And that's it. So one of the last major advancements that have been made recently with DNA biotechnology is actually the sequencing of DNA. And it really just started with sequencing certain genes. I think the first one was sequencing insulin, and they actually started with the protein. But by sequencing these genes, they can then study mutations and how the genetic code towards the proteins works, and it just helps to have that information. It eventually led to the sequencing of entire genomes of just all the genetic material of an entire organism, which that's just a huge amount of data, and we'll actually be getting into genomes in our next video, so I'll save some of that. In this slide, we're going to talk about DNA sequencing, and essentially the most common method that is used is called dideoxy chain termination method, and you don't really need to know what that means. Dideoxy is, as opposed to deoxyribonucleic acid, there's a further chemical change that's going to be used, and chain termination is going to be at the end of the chain. Okay, so basically, you'll have fluorescently labeled DNA molecules that are chemically altered, like I mentioned, and this chemical alteration will actually make it so that it can't bind on the 3' end, only on the 5' end. So wherever this altered DNA molecule, or wherever this altered, it'll actually be just a base pair, wherever this altered base pair is, will be the end of the DNA chain. You can no longer have any more bind on the other end. So first what they'll do is they'll prepare many copies of the strand, but in the presence of these fluorescently labeled altered base pairs, so that each chain that's created will terminate with that specific labeled base. In this, they're just kind of color-coded. You could have it labeled with fluorescence, so that under UV light, each one will have a different wavelength. Or maybe they'll use radioactivity with just differently distinguishable radioactive signatures or whatever. But each chain that's created will terminate with a specific one of those labeled bases, and the molecules can then be sorted in a process similar to gel electrophoresis, but the molecules can be sorted by length, and it's precisely enough that a laser can recognize those labels, those fluorescent or radioactive labels. And you can just kind of see in this diagram here, each one is only slightly longer than the previous one, but there is no way to mix them up. They're completely distinct lengths, and so by separating them by their lengths, you can then read the fluorescent label just one at a time in order to see the sequence. And so this process was largely automated over time, rather than just being read by hand. So once they had those machines reading that, the DNA could be sequenced efficiently for both genes and for genomes. And so that's really it for biotechnology, for DNA biotechnology. It's just as simple as that.